at any moment in time, I might be more of an artist, or I might be more of a scientist. In fact, I could sit here and talk about this as either a scientist or an artist. This is a nucleosome structure. There's at least 3,000 welds in this, and it's composed of DNA, which is shown here on the outside, and proteins in the middle. So the histone proteins are shown in the middle, the DNA is shown on the outside. If you think about that, we have to package all of that very tightly into each cell, and that's one of the functions of this protein DNA complex, is to do this packaging. And in fact, I even made the entire DNA structure here and ended up tearing it down and rebuilding it, because it wasn't accurate enough. And I'm giving this to the woman who discovered the structure, so it's got to be as accurate as I can make it. I was able to complete this with some of my colleagues here. Brad Cairns helped a lot with this project, and I'm going to be giving this to her next week as basically to honor and recognize her for her accomplishments.
It's just so tactile, and you interact with it in a more, I think, a deeper and a more intimate way. When you can touch it and you can feel it and you can look at it, it's 1/50 the size, as you mentioned, in real life. But it's a real physical entity too. I notice empty spaces and interactions in places that surprise me. I mean, DNA is supposed to be a rod, and it's actually kind of hard to bend.
Our goal is to have many of these structures here at HCI, and also have information sheets that we can provide to people that will describe the structure, and also describes the research that's going on here based on the structure. We have fantastic scientists here that are actually studying the nucleosome and the histones and how they regulate cancer.